I will be talking about today about dissecting and predicting bull fertility. And in my talk, I will discuss some of these points. Does genetics affect bull fertility? What's the relevance of non-additive effects? I will talk about a recent study about the identification of major causal variants. And I will talk about prediction. Can we predict bull fertility using genotype data? I will discuss about the use of biologically informed genomic models. And at the end of my presentation, I will talk about the use of a cross-country reference population for predicting bull fertility. My group has been working on dairy bull fertility, but the rationale of our research, the questions, the methods, and even the findings can be directly applied to bull fertility in beef cattle. So what's the importance of bull fertility? You know, improving reproductive efficiency is a major goal, not only in dairy cattle, but also in beef cattle. Fertility is an extremely important economic trait. And despite recent advances, the reproductive performance remains suboptimal. I have some numbers here from Holstein in US. We have like a 400 days calving interval, more than 2 services per conception, and 114 days from calving to last breeding. You guys can hear me well? Yes?
Yes, sir.
Excellent. So you will agree with me, most studies have focused on cow fertility, either dairy cow fertility or beef cow fertility, while bull fertility has received less attention. And this it's a bit contradictory, given that we can use one bull to mate many cows, and then one subfertile bull will have a larger impact on her fertility than a single cow with fertility problems, right? We know now that service sire affects not only the fertilization process, but also early embryo development. And recent studies have shown that a significant percentage of reproductive failure is attributable to bull subfertility. Now, how do we evaluate bull fertility? Well, traditionally, we have been evaluating bull fertility using sperm traits like sperm count, sperm morphology, sperm motility. And in beef, it's common to perform a breeding soundness examination or use scrotal circumference. Now, alternatively, we can evaluate bull fertility using field records, right? We can use conception rate, pregnancy, check records to evaluate bull fertility. And I will argue that sperm traits or breeding soundness examination or scrotal circumference, those are indicators of bull fertility. Those are proxy traits. But if we want to measure the true fertility of a bull, we should measure it in the field using conception rate records. Now, the dairy country in this industry has access to a phenotypic evaluation of dairy bull fertility since 2008 called sire conception rate. Sire conception rate is a phenotypic evaluation of bull fertility adjusted for variables related to the cow that received the unit of semen. So those nuisance variables like lactation, age of the cow, milk production, days in milk, all of those nuisance variables are removed. And in that way, we have an adjusted phenotype for bull fertility. This is data from a few months ago, December 2020. At that time, we had 15,000 Holstein bulls with roughly 72,000 SCR records. So we have multiple SCR records per bull. You can see here the distribution, beautiful graph. SCR records follows a long tail bell-shaped distribution. The mean by definition is equal to 0, but you see a beautiful variability with more than 10% conception rate difference between what I call, I hope you can see the pointer here, high fertility bulls and low fertility bulls. Please note that all these bulls are in the market. That means all these bulls have good, very good or excellent semen production and semen quality trait. So even when the bulls have excellent semen trait, then in the field you observe some quite important differences in the fertility of those bulls. So some years ago, we asked ourselves this following question: does genetics affect bull fertility? And we try to answer this question using data from 11,000 Holstein bulls with more than 45,000 records of bull fertility. So we have multiple records per bull, and we have roughly 70% of the bulls with genotype data. So we perform a single step analysis where we could combine all the phenotypic, all the genotypic, and all the pedigree information. So in this plot, a classical Manhattan plot, I'm plotting the amount of genetic variance explained by small windows across the bovine genome. And if you ask me, I would say, well, bull fertility in Holstein, it's a typical polygenic trait affected by multiple genes with small effects. Now, if you carefully evaluate the most prominent peaks, you will see that most of these peaks harbor genes that are directly related to bull fertility, including spermatogenesis, sperm motility, and the fertilization process. We did the same kind of analysis, but using data from Jersey. We had 1,600 Jersey bulls, again, multiple records per bull. And we have, in this case, almost 80% of the bulls with genotype data, but we perform, again, a single step analysis. Same picture. Bull fertility in US Jersey breed is a polygenic trait. Many multiple genes with small effects, no window across the genome explain more than 1% of the genetic variance. And again, if you carefully evaluate the most prominent peaks, you will find that most of these peaks harbor genes associated with male fertility, including testis development, spermatogenesis, and the acrosome reaction. So what have we learned about these two studies? Well, it seems genetic factors explain part of the variation in dairy bull fertility. Most of the significant regions genes are breed specific. That is a very cool result, and this could be due to different reasons. Different causal mutations are segregating in Holstein and in Jersey. Or it could be that the same mutations are segregating, but they have differences in the LD pattern. That means the mutations are segregating in Jersey, but they are not in high linkage disequilibrium with the markers that we have in the commercial SNP chips, or could be just false positive, false negative results. A very good result is that if we evaluate pathways or molecular mechanisms underlying the most significant genes, we will see that these pathways are common to both breeds, and this provides further evidence that pathways, mechanisms rather than single genes are the primary targets of selection. Now we have to acknowledge that in these two studies we evaluate additive effects, but we have other non-additive effects, dominance, recessive, and overdominance. And it's believed, at least based on the theory, that non-additive effects are important for traits related to fitness, such as bull fertility. So we ask ourselves, what's the relevance of non-additive effects on bull fertility? And for that, we use data from almost 8,000 Holstein bulls. In this case, we use one record per bull. The most accurate one is the record that is based on most breedings and roughly 60,000 SNP across the genome. And we found 4 major regions on the bovine genome that has very significant non-additive effects on bull fertility. As you can see here in this Manhattan plot, each point now is a SNP, and I'm plotting in the y-axis the observed minus log 10 p-value, and you can see clearly 4 major regions, one on chromosome 8, one on chromosome 9, one on chromosome 13, 13 and one on chromosome 17 that show very significant dominance effects. If you check the size of the p-values, we are talking about p-values at -15, -20. So we were very excited with this result, and we decided to validate this result using a larger population. In this case, we are using 12,000 Holstein bulls and much more markers. We are using here 300,000 markers across the genome. The idea was to perform a fine mapping. Again, each point here is one marker. We found exactly the same 4 regions, one on chromosome 8, one on chromosome 9, one on chromosome 13, and one on chromosome 17, and a new one on chromosome 27. A beautiful Manhattan plot. You see a very, very neat peak in each case, and please, you can check in the y-axis the how significant are these regions. We are talking about p-values at -30 and -40. Some outstanding results. And here I'm showing the distribution of bull fertility per region. So we have the most significant marker, 2 alleles, 3 genotypes, and you can see clearly here that we are talking about a dominance effect, where bulls that are AB or BB, they have the same performance, where bulls that carry the AA genotype, they are somehow subfertile, right? With some very extreme examples like chromosome 27 or chromosome 9 or even chromosome 13. So we decided to identify the causative mutations that are underlying these major non-additive effects. And for that, we perform a target sequencing of each candidate region. We sequence 10 megabases per region. And actually, we designed per region 2 pools. One we call the low fertility pool. That was a pool with roughly 25 bulls, all of them with genotype AA for the most significant marker, and a sire conception rate on average -7, quite subfertile animals. And then a high fertility pool, again, 25 bulls, all the bulls with genotype BB for the most significant markers, and those are high fertility bulls with SCR around +5. Here is our wish list, and actually we decided to perform a conservative approach. So we wanted to detect either missense or nonsense mutations. We wanted to have opposite homozygotes between the low and the high fertility pools. If we were talking about missense mutations, we would like to have a very deleterious effect. And of course, we would like to have genes that carry missense mutations, and those genes either exclusively or highly expressed in testes. We published this study just a few weeks ago in Animal Genetics. Let me show you a couple of examples. This is what we found on chromosome 9, a very significant region. We found a missense mutation in AK9. That's an enzyme that is involved in nucleoside homeostasis. And you can see here the expression profile of this gene using mouse tissues, and this gene is exclusively expressed in testes. This is another example. This is chromosome 17. This is a missense mutation in FOXN4. This is a transcription factor. This mutation is very deleterious. It's the change from a polar to a non-polar amino acid. So low fertility bulls carry a very deleterious mutation. And you can see the expression profile of this transcription factor using data from humans. This transcription factor is exclusively expressed in testes. So we have identified what we call strong candidate causal variants in each of these regions. We are now phenotyping these mutations. The idea is to try to see if the mutations affect either fertilization or early embryo development or even elongation, and probably in the future we will need to run some gene editing experiment in order to validate. And this result could have a major impact on the dairy industry, because if we include these markers on commercial chips, we will be able to screen newborn bulls and exclude those, discard those, call those that will have low fertility, right? Now, before I talk about prediction, let me show you this study about homozygosity and bull fertility. We know that inbreeding affects performance, and inbreeding depression tends to be more important on fitness traits, such as bull fertility. So inbreeding or the loss of genetic diversity can be quantified using either pedigree or genomic data, or can be also quantified using runs of homozygosity. This concept of using a long stretch of homozygous genotypes in the chromosome of a given individual. And that's what we use here. We evaluate runs of homozygosity. Here in the first plot, A, we have the distribution of SCR record for roughly 12,000 Holstein bulls, and we focus on the two tails, the top 300 high fertility bulls and the bottom 300 low fertility bulls. And for those bulls, we compare the runs of homozygosity. Plot B, you have the average size of the runs of homozygosity, and in plot C, you have the total size. And what we found is that both the mean and the total length of run of homozygosity were greater in low fertility bulls. In other words, low fertility bulls tend to be more homozygotes, tend to have lower genetic diversity. What we also did is evaluate the enrichment of runs of homozygosity regions in low fertility bulls. We did that using a Fisher-Exact test. So for a given ROH in the genome, we tried to see if that ROH was enriched or was overrepresented in the low fertility group. And actually, we found that 9 regions across the genome that have ROH are all enriched in low fertility bulls. Some of these regions harbor actually genes very related with bull fertility. So probably the most important finding of this study is that homozygosity might be an important risk factor for bull subfertility. In other words, the loss of genetic diversity can be a risk factor for bull subfertility. Let me talk about prediction. You know that prediction is relevant nowadays, not only in animal and plant breeding, but also in personalized medicine. And the goal is to predict either genetic values or phenotypes. And accurate prediction can be extremely challenging, right? So we ask ourselves, can we predict bull fertility using genotype data? And we try to answer this question using data from more than 7,000 Holstein bulls, using 61,000 SNP across the genome. And please focus here on the left, the white black box where it say all, that is using the 61,000 SNP, and we got a predicted correlation in cross-validation of almost 35%. Beautiful result. So we evaluate predictive performance in cross-validation. Remember, that means we feed the model, we train the model in a given data set, and then we evaluate the performance of that model in a completely different data set, and then we compare the prediction with the fertility that we observe using a correlation. So as I mentioned, this 34, 36, 35 percent, it's quite good just to compare. We can get the prediction accuracy if we do some massages, like predict the correlation divided by the square root of the heritability. If we do that, we get a prediction accuracy of 0.62. Again, just to compare, in Holstein for postpartum health disorders, we have prediction accuracies around 55%. And the prediction accuracy for feed safe, this novel trait that now we can use to select for feed efficiency, that trait has a prediction accuracy up to 40%. So again, this 50 to 62%, sorry, prediction accuracy for bull fertility, it's an outstanding result. So if you read papers about bull fertility, you will find that some authors think that bull fertility is affected almost exclusively by environmental effects. So basically, genetic has nothing to do with bull fertility. So we were saying, how come we can predict bull fertility in gross validation using the genotype data? In other words, are we missing some hidden variables that are actually driving this result? So we did this very cool exercise using the Jersey data set because it's smaller, and we perform a permutation analysis. In the permutation analysis, what you do is you break the connection between the phenotype and the genome of the bull. You shuffle genotypes across phenotypes, you break the link, and then you try to predict bull fertility using genotype data. You don't do it once, you don't do it 10 times, 100 times, you do it thousands of times. So when you do this, you get a prediction correlation equal to 0. That means when you break the connection between the genotype and the phenotype, now you cannot predict bull fertility. And that means that the predicted correlations of 30% that we get by, in Jersey are completely driven by the genotype of the bull. What a beautiful result. So recently we evaluate two things. We evaluate the importance of using the X chromosome. And as you can see here in blue, 0.32 to 0.34, or here in orange, if you include the X chromosome in your model, you will get more accurate prediction. That's a good result. And the second thing that we did is to evaluate the importance of including non-linear effects. So here in blue, we are using a linear kernel model. No worries about the name. This is a classical GBLAC model. And in orange, we are using a Gaussian kernel. It's a model that can capture non-linear effects from genome to phenome. And what we got is that if you want to predict bull fertility, it's better if you use non-linear effects. What makes sense, because we are trying to predict a phenotype, and if you are trying to predict a phenotype, it's better to include more than just additive effects. So we have been working with biologically informed genomic models. Genomic prediction works. It's outstanding. There is no discussion, and has caused the most remarkable change in dairy cattle breeding since the introduction of artificial insemination. Now, genomic prediction is a black box tool, right? Genomic prediction ignores any available information about functional features of the genome. So we believe that the use of prior biological information can improve two things: predictability and also model robustness. And model robustness in special could be something very important in beef cattle breeding. There are different types of biological information that you can include in a model, like pathways or gene sets, major mutation or functional annotation data. Let me show you in the next slide some of these examples. I showed you this data before. These are these 5 major massive non-additive effects that are segregating on the genome of Holstein bulls here in US. So we play with these mutations for prediction. So here in yellow, we have what I call the base model. The base model is a model that is fitting roughly 300,000 markers across the genome. And with that model, we get predicted correlations in cross-validation around 35%. Now, when we extend that model, and that is green, we extend that model by removing the most significant markers from the G matrix and fitting those major markers a fixed effect. then we achieve predictive correlations up to 40%, and you can see here, and we also reduce the mean square error of prediction. So these major mutations are real. They have a major impact on bull fertility, and if we model those mutations in a predictive model, we can get more accurate predictions. So here we try to predict bull fertility by using functional data. So we use different classes of functional data. We use synonymous mutations here. We use non-synonymous mutations here. We use mutations on regulatory regions. And for each class, we compare two models: a model fitting functional variants and a model fitting the same number of SNP that the model fitting functional variants, but SNP at random from the genome. And the idea is basically to separate the population structure that, that the markers can explain from the biological roles. And as you can see here, in blue are the models fitting functional variants, and in light blue, the models fitting random SNPs. The functional variants exhibit higher predictive power than random variants. And that is easy to see in the non-synonymous, the difference is 5%, and somehow with also synonymous mutations. Very cool result nowadays that we are moving to use whole genome sequence data, how we can filter these 30 million SNP that we can get in order to boost genomic prediction. Lastly, let me show you this recent study where we performed within breed across country genomic prediction, and you know, for some small breeds and traits that are difficult to measure, across country reference populations could be a very cool idea, right? And we performed this study in Jersey. In Jersey, we have 1,500 Jersey bulls with bull fertility records in US. And in Australia, we had access to 600 Jersey bulls with semen fertility values. These traits are somehow different, but are correlated. The genetic correlation is roughly 50%. And let me show you the result here. In blue is the predictive correlation within country in Australia after we could optimize the reference population, and we could get predictive correlation around 20%. But the best performance was achieved with a bivariate model, that is fitting the entire US data set and the Australian data set, considering this trait as different traits but correlated. And in that case, we got predictive correlations up to 30%. And actually, we did a remarkable we achieved a remarkable improvement in the ability to predict bull fertility in Australia from using all the records to only use the most accurate one to try to define, optimize the reference population, and we end up in a bivariate model where we use information from the records from US. Very cool result. This is my last slide. Let me finish talking about this beef on dairy, because there is now a great opportunity to evaluate bull fertility in beef cattle using dairy cow records. For that, let me show you this recent study. This study is from Daniela Lorenzo and her group in the University of Georgia. So Daniela and her group evaluate the sire conception rate of Angus bulls using dairy cow records, Holstein cow records. Let me show you here in the first row, the mean, the number of services per bull was around 1,500, but let me show you here, as you expect by definition, the mean is 0, that's by definition, but look at the variation. So we have Angus bull that we can consider high fertility, but we have some low fertility bulls. And this is a unique opportunity now to evaluate the fertility of beef bulls using this data that come from dairy farms. Some take-home messages. Yes, genetic factors explain part of the variation in bull fertility. Probably one of the most important findings is there are major variants with large non-additive effects affecting bull fertility. That is not only true for Holstein, but it's also true for Jersey, and it's also true for Brown Swiss. So it's true for different dairy cattle breeds. And I don't know the answer in beef cattle, but that will be something that we should investigate. Another important topic is the homozygosity might be an important risk factor for bull subfertility. The loss of genetic diversity can be an important factor for bull subfertility. Genomic prediction of service sire is feasible, and that is a great opportunity for accurate genome guide decisions, such as early culling of newborn bull calves with very low fertility predictions. The use of functional information can improve model predictability. This idea of using across-country evaluations could be good for small breeds, but the use of across breeds could be also an opportunity for some small breeds in beef cattle. And finally, beef on dairy. This represents a great opportunity for evaluating beef bull fertility using data that come for free from dairy farms. With that, let me thank my team. The old members, the current members, they deserve all the recognition. They do all the work. I'm just the cheerleader of the group. And let me thank the data providers and also the money providers. With that, thanks for your attention. And of course, I'm open for questions.
Thank you, Dr. So we'll start with questions from the audience. We might have to come up and ask the question too, so we can make sure that we hear. I'll start with the question. So Dr. Francisco, the research that was reported by Dr. Olney, how reliable would that be to translate into the beef side of it, maybe more particular, the permutation analysis that you talked about in the following study with these, at least Angus bulls we have out here, or other beef bulls we have low AI utilization, and how you can collect those records?
Yeah, your question is if how we can use this methods on beef cattle where we have low AI records, right? Yes. So what I'm thinking is, well, based on the study of Daniela Lorenzo and her group, first, it's possible to have access to a large data set from dairy farms, right? Those are all AI records from Angus bulls. The discussion, and I think it's interesting, is maybe those Angus bulls are not elite bulls for Angus breeders, so they could be somehow a dissociation there. But also we should have access to AI records from Angus breeders. So then we can merge these two data sets, create a unique database, and then try to predict bull fertility using genotype data. The beauty of that is that you will have a prediction for a newborn bull, and you don't have to wait until the bull is out in the market or in the field to realize that he has low fertility, right? The other major finding, I think, in dairy cattle is the segregation of these major non-additive effects. And that should be something that we should investigate in beef. And after we capture those effects, we can track those using SNPs and try to either remove the alleles from the population or at least avoid the homozygote bulls, right?
Thank you. We have a question from the audience.
The correlation between scrotal size and early heifer maturity reaching puberty.
Did you hear that question? The correlation between scrotal circumference and heifer puberty.
If I know the answer, no.
Well, that was a good honest answer. Other questions from the audience?
Now, let me say something that maybe can help, at least in dairy. The correlation between male fertility and female fertility is almost zero. That means the correlation between the fertility of the bull and the fertility of the daughters of the bull is almost zero. So we are talking about almost two uncorrelated traits.